Hello students, I am Khedkar N.D from RBNB College, Srirampur. I will start the biologist topic, that is reproduction in lower and higher plants. The hints are given to solve the questions of CET and NEET. The next point is that is TSOP and THER. TSOP and THER. You know that anther, anther consists 4 pollen sacs. Four pollen sacs like this. 4 pollen sacs are present. In each pollen sac, microspore mother cells are present. From the microspore mother cells, pollen grains are developed. Pollen grains. From one microspore mother cell, 4 pollen grains are formed by meiosis. Then, in the wall of mature anther consists 4 layers. आता हे झालं मायक्रोस्पोर मदर सेल्स बद्दल. त्यानंतर wall of the microspore mother cell consists the 4 layers. Epidermis, endothecium, middle layer, epidermis, middle layer, endothecium, middle layer, tapetum, likewise the 4 layers are present, and both these layers are perform different functions. या पॉइंट वरती देखील प्रश्न विचारले जाऊ शकतात. एपिडर्मिस, function of epidermis, where the epidermis is present. Endothecium, that is hygroscopic in nature, and hence it is useful to burst or rupture the pollen sac. pollen sac rupture करण्यासाठी म्हणून एंडोथिशियमची मदत होते. Epidermis, that is protection and outer layer of epidermis. Middle layer at the time of maturity it get disappear and both the pollen sacs are mixed with each other. And this is the function of this middle layer. Hint for these lines that is layers, function, role. Tapetum is a nutritive tissue. It supplies the nutrition to the developing microspores. Here like this tapetum is present and they are nutritive in function, developing sporogenous tissue or developing pollen grain. It supplies the nourishment. About this, the structure of the TSOP pollen anther. Then microsporogenesis, that is formation of pollen grains, that is microsporogenesis. formation of pollen grain. पोलन ग्रेन तयार होण्याची जी प्रोसेस आहे याला मायक्रोस्पोरोजेनेसिस असं म्हणतात. स्ट्रक्चर ऑफ पोलन ग्रेन. स्ट्रक्चर ऑफ पोलन ग्रेन. स्ट्रक्चर ऑफ पोलन ग्रेन. मग आता यामध्ये पोलन ग्रेनच्या स्ट्रक्चरवरती देखील प्रश्न विचारले जाऊ शकतात. How many germ pores are present on the pollen grain? How many layers are present on the pollen grain? How many pollen grains are formed from a single microspore mother cell? नहीं का? Cytoplasm. What is the outermost layer of the pollen grain? Name of the outermost layer of pollen grain. That is XII. Name of the inner layer of pollen grain. Intine. What is the function of XII? XII, it is outermost layer, and that is sporoderm. Sporoderm is the layer of pollen grain, and outer layer XII is thick and made up of complex non-biodegradable substance, that is sporopollenin. Most important word in XII, sporopollenin. Sporopollenin is present, and it is non-biodegradable. Non-biodegradable means any bacterial action, any microbial action are not takes place on the pollen grain, that is on XII. म्हणजे या ठिकाणी पोलन ग्रेनचं प्रोटेक्शन हे स्पोरोडर्म मुळे होऊ शकतं. जमिनीमध्ये पोलन ग्रेन हा राहू शकतो. मग जर आपल्याला याच्यावरती प्रश्न विचारला, नेम द केमिकल सबस्टन्स प्रेझेंट इन द XII, त्यामध्ये आपल्याला पर्याय वेगवेगळ्या प्रकारच्या देतील, पण त्यामध्ये आपल्याला स्पोरोडर्म दॅट इज स्पोरोपोलिनिन हे आन्सर त्या ठिकाणी करेक्ट सांगावं लागेल. ओके. Development of male gametophyte. Next point, development of male gametophyte. Male gametophyte. From the microspore or pollen grain, the development of male gametophyte takes place. You know the structure of male gametophyte. XII, intine. Names of the layer XII, intine. Then here the nucleus is divides into two cells. That is, pollen grain is divided into two cells, generative cell and vegetative cell. Vegetative cell and generative cell. Generative cell, vegetative cell. Then here from the generative cell nucleus, the two male gametes are formed. Next step is that it divides mitotically and two male gametes are formed. From exine, the pollen tube, pollen tube is formed, and from the generative cell, two male gametes are developed. In this way, these two male gametes are non-motile and developed from, that is, generative cell flies into the cytoplasm of vegetative cell. Second mitotic division is concerned with the generative cell only and gives rise to two non-motile male gametes. अशा पद्धतीनं दोन non-motile male gametes हे generative cell पासून तयार होतात. From generative cell. Here, how many male gametes are formed in the development of male gametophyte? Question is asked on this point like this: how many male gametes are formed? That is, two male gametes are formed. These male gametes, the formation of male gamete is takes place in higher plant. These male gametes are motile or non-motile. That is, these male gametes are non-motile. They cannot move because they are carried by the pollen tube during fertilization. अशा प्रकारची हिंट आपल्याला इथं सांगायची आहे. Then the another point, gynoecium, that is female reproductive organ, gynoecium. Gynoecium, female reproductive organ of the flower. Individual member of gynoecium is called the carpel. It consists, carpel consists stigma, style and ovary. Stigma, style and ovary. Stigma is a receptive part. A receptive part means they receive the pollen grains. The pollen grains are received. Style is give the way to the pollen tube. When pollen grain germinates and gives way to the pollen tube to pass towards the ovules. Ovary is the female organ of the flower in which ovules present. ओव्हरी मध्ये ओव्हुल्स असतात. अशा पद्धतीनं आपल्याला गायनोशियम बद्दल द फिमेल गॅमेट म्हणून सांगता येईल. स्ट्रक्चर ऑफ अनाट्रोपस ओव्ह्यूल. Structure of anatropous ovule. In the ovary, the ovules are present. In the ovary, ovules are present, and the structure of this ovule is three antipodal cells, two integuments, outer integument, outer integuments. They perform the function of protection, and as well as after fertilization from outer integument testa is developed. Totally, that is after fertilization ovule get converted into seed. ओव्हलचं रूपांतर सीड मध्ये होतं आणि मग आउटर मोस्ट लेयर जे आहे याचं काय होतं तर या ठिकाणी आउटर मोस्ट लेयर जो आहे यापासून टेस्टा डेव्हलप होतो. टेस्टा इंटुगमेंट्स, इनर इंटुगमेंट्स, from inner integuments the tegmen is developed. Here egg after fertilization from the egg zygote, and from the zygote, that is oospore, from the oospore, that is embryo, female gametes. Secondary nucleus after double fertilization develops into the PEN, that is primary endospermic nucleus, and later on endosperm is developed. Then question is asked on this point, the different functions of outer integuments, inner integuments, or after double fertilization the secondary nucleus is get converted into which part, that is endosperm. Function of outer integument is protection and formation of tegmen. Function of inner integument, that is tegmen. Outer integument, that is testa. Inner integument, tegmen. Then from egg, that is embryo is developed. Female gametophyte, actual female gametophyte is developed from the egg. Okay. One more question is asked down here also. Funicle, here with the help of funicle, it is attached to the placenta. Ovule is attached to the placenta with the help of funicle. Placenta is this part supply to the nourishment to the developing embryo. प्लासेंटा मधून फूड सप्लाय केलं जातं कुणाला तर डेव्हलपिंग एम्ब्रियोला, आणि मग हा प्लासेंट्याला अटॅच फ्युनिकलच्या मदतीने, फ्युनिकलच्या आधारे अटॅच झालेला असतो. फ्रॉम विच पार्ट द ओव्हल इज अटॅच टू द फ्युनिकल, दॅट इज विच पार्ट ओव्हल इज अटॅच टू द प्लासेंटा, दॅट इज फ्युनिकल. हे आन्सर आपल्याला इथं सांगावं लागेल. आफ्टर फर्टिलायझेशन दॅट इज ओव्हल गेट कन्वर्ट इन टू सीड. ओके, इथपर्यंत आपण बघूया, इथं थांबूया. ठीक आहे.